My name is Wedad Fallatah, and it has been a pleasure. I've been here for the last 3 years at Kennedy Krieger, and it was a very valuable resources in term for me to develop and learn so much about the adult refsum disease. It's actually, this is the beginning of the work group. We haven't formally started the work group, so I am just talking. It's kind of building the things together, and I'm very excited because now we see all the other groups and what they are doing, and I think it's kind of summit what we have and what we can use in the future. So let's get started. So first of the question, so I know this is maybe like kind of repeatable, but if we are thinking about it in a very basic way, why the research infrastructure matters, especially in adult refsum disease. And when I look at this, I found it's very good for me to just summarize it in 6 core functions that help for research infrastructure rather than thinking about it or buildings or database. And it's kind of a foundation of every stage of our way or our journey in this disease. So everything, if we can see, it's actually began, and now we know it's important by learning today that everything it's began with understanding the disease itself. Natural history studies allow us to define the disease progression, understand the variability between patients, and identify meaningful clinical outcomes. And it's also helped us to establish the baseline against which the future therapies can be evaluated. The second, it will be the biomarker, as we all discussed in the previous groups, and how important to accelerate the biomarker discovery, as these are essential and monitoring the disease progression. And I like the talk today because a lot of researchers found that it's not enough only to look at the phytanic acid, but it could be there are other biomarkers that could help us to move the research forward for ARD. The third for these, the infrastructure, it will be talking about the model, and that's what I will touch base later. And here you have the experimental models that help us to move the clinical observation and to understand the disease mechanism and the pathophysiology. Today, I will talk briefly what I learned here by working with a whole group of the peroxisomal expert in the disease, and what we develop in terms of the mouse models that we are working with. And then the fourth one will be the enable us to therapeutic discovery. And here, once we have all these kind of foundation in terms of the natural history data, the model, this is kind of help us to develop drug screening, targeted validation, and translational research that bridge the laboratory discovery that we have to the patient care. And this will move us toward the build trial readiness data set and the infrastructure, as even that's the most promising therapy cannot reach without the trial ready infrastructure. So these are all kind of discussions that we are all enjoying and participate. It's very important actually in the future. And this will help at least to discuss the standardized outcome measures, harmonized protocols, and share all the registries. It also will strengthen the international collaborations to support multi-center clinical study. And eventually that will lead to the to the global community connection. And this is perhaps most importantly, research infrastructure that connects all patients together and also connects researcher expert in the field. Together we create this research system that will be much stronger than a single institution. So let's move on. So let's talk a little bit what we are today. And I know that so many talks today. So by the end of the day, you all have this kind of great background, what we actually are, where we are in, and what's the current challenges that still like try to focus to improve for this condition. So adult refsum disease research has made remarkable progress over the past few years, thanks to the Global Dare Foundation and the expert and the patients for sharing all the community. And this is important because now we start to have these kind of building blocks, and we have the clinicians with deep expertise, the researcher, they're willing to study, and they're already started the patient advocacy driving these kind of collaborations. The natural history data now start like building up, and this will help to emerge more biomarkers and also let us think about what's the valuable biospecimen. As Christy mentioned before, maybe like we will look for the dry blood spot, the whole blood, other thing that's easy for patient to collect, in addition to the experimental disease model and the global, as I mentioned before. However, we still have a lot of challenges because it's a small community, so it's very hard to find sometimes the patient, and it's very hard to have the whole number or set of patients to do the trials or the study. The longitudinal data sets, still we have a limited knowledge about that. We have a limited knowledge about even the data basic for this condition, and also the standardized outcome measure that will help us to actually look for a measurable outcome for this condition. This is very important to think about as we are building this research infrastructure. We're still missing a biomarker, and this is all kind of, I think now I see all the talks, I'm very hopeful that few years from now, we will have all these kind of building blocks that we were talking about today. Okay. So this is the framework, which I think it's all started from the patients. The patient is like the center of this, and we learn so much from them. And sometimes the patient, they are expert, as I can see for this group. With this work group, I think it's very helpful for me to just look and see a connected research system in which we will have the clinical phenotyping and connect that with all the data that we try to learn in terms of the biospecimen that we use, multi-omics, lipidomic, and other omics data set, biobank, data science. So collect all these kind of things together and put it in a way that's helpful, not only for the researcher, but also for the future to help us for the preclinical testing, for drug discovery, and for clinical trials. Okay. And from that, I will move a little bit to talk about the experimental models and what we can, as a group, think about it. We know that we have, sometimes we have the fibroblast, and that's one of the kind of an important discussion. So these are the patient-derived fibroblasts are a valuable tool to study this condition. In addition, we can think about the iPSC, organoids. So these kind of all just the brainstorming idea. Now we know about the mouse model, but as the disease like moving forward, we have to think about other models that we can use, especially that I'm learning today about the retinopathy and how is this affecting patient. We really need to focus on this and try to explore other ideas that can help us for this to understand this condition better. Okay. From that, I will not explain a lot, I know. So I think I'm the junior here, so most of you are expert for this condition. The only thing I want to point out from this, because I missed some of the talk in the morning, but I think the point about phytanic in the diet and how is the dietary management for phytanic is important. But thanks to Anne Moser, I also learned here. So I'm very grateful for the group here and Dr. Paul Watkins. So I learned that not only the phytanic acid, actually now we are seeing that the precursor, the other dietary precursor, also contribute to the phytanic acid pool, and those namely the phytol and the phytol fatty acid esters. So these are compounds are a storage form in the phytol, and these are found in the plant tissue, particularly during the chlorophyll degradation. Although the PFAE have been recognized biochemically, their contribution to phytanic acid accumulation has remained poorly understood, and this knowledge gap become one of the major motivation for us to look at that for this condition, particularly when we see a lot of patients, they are kind of well controlled sometimes with the phytanic acid diet, but still they have this high level of phytanic in their blood, and this is might be now we are studying it. Maybe this is kind of help us to understand a little bit about this condition and the contributions of these other precursors to the whole picture of the elevated phytanic acid in ARD. Okay, so I think that's all, and that's the point I need to import here. Yes. So with that said, we have been, so I have been part of the group. So this is a collaborative work in which a group of the researcher in the Johns Hopkins established a novel mouse model in which we were able to knock out the phi-itch. And those mice, they were on pure black C5 C57 Black Sea background. And this was exciting because the existing animal models, if you look at the literature, the existing animal model or the previous one that published developed significant toxicity and weight loss when it comes to the challenging with the phytanic acid diet. And there are other models, such as also the PEC7 that can use, but however, that exhibit a broader peroxisomal dysfunction, which is make it difficult to just isolate the effect of phytanic acid accumulation itself. So that's kind of motivation for us to study this model. And that's when I came to the lab to learn all about how to measure the phytanic acid and how to prepare the diet. So what we did here, so we have a baseline collection of blood sample from the mice, and we have different kind of group of mice. We have the wild type, and we also have the knockout model, and we give them either the standard diet or a various a kind of dose dependent or a various concentration of phytanic acid. And then we measured the blood before starting this feeding regimen, and then at the midpoint of the study, we also measured the blood for phytanic acid by GCMS mass spectrometry, and then at the end of the study, in addition to collect the blood, we also collected the tissue, and those are the one that we just mentioned previously about the importance for looking for the retina. So maybe those the eye tissue can help us to look for the pathology in the retina in terms of mutant under standard and mutant under a high phytanic acid condition. And I hope that will be a valuable resource for the community. Okay, let's see. So this is kind of it's an ongoing, but hopefully we will have completed picture of the data by the end of this year. So on the top, when we look at the phytanic acid in the blood, and here is as the baseline, and you have the different groups that we are measuring, and then we see a dose-dependent accumulation of phytanic acid over time. And as you can see, by the end of the study, we see this higher level, and it's also matched the higher concentration of phytanic acid. Interestingly, so we also see that more, so it's variable in terms of tissue. When we look at the tissue, we see some variation in the elevation of phytanic acid compared to the blood. Interestingly, if you look at the right side of the slide, recently when we looked at the precursor, which is the phytyl and the phytyl fatty acid esters, we also see an accumulation, and this is accumulation. So we have at the midpoint, but now we also have at the end point, and we see this kind of accumulation over time. So not only the phytanic acid, also the precursors contribute to this elevation. And on the bottom, when we look at we measured our food as a validation that the food is actually contain this kind of specific measurements of the phytanic acid, and we see that confirmation when we look at the chow diet. Okay, so these kind of it's a very important representation that it's kind of and the phytyl fatty acid esters underappreciated before, but it could be a major contribution, and this is important for dietary management and also for further investigation for human studies. Okay, so let's go. So that's kind of a case study. That's what we did in the lab. And Muzer, she's also doing the food testing program, which is another thing. I didn't talk about it for the matter of time, but that's another important, I would say, important skill that I learned during my years here at the Kennedy Krieger. Okay, so now we move toward translational therapeutic development roadmap for the ARD, which is kind of the important thing that we have to, as a group, think about and plan ahead from now about that. So far, we've discussed these all kind of a research infrastructure, the connected research system that can help, the experimental models, potential models that we can look for. So how we can translate these discovery into therapies to improve the life for patients. And I think when I look in my mind, I look at those as kind of tiers. So which kind of things that's available or feasible for us to work on now versus those can take time just to develop and to look for the logistic and to look for the funding. And as you can see, I just categorize them. So I think maybe the first thing that we have to look is the dietary management. Now we look at that, and maybe I didn't put here the retinopathy and the retina, so maybe this is like the second important things for the patient to look for and to study, particularly that we have now the mouse model that can help us to evaluate this pathology and to learn more about this condition. And then you have things that can needs more time, but it's very valuable for us to, at this level stage, to discuss these kind of point or with the goal in mind to translate these kind of discovery and translate it to something very helpful in the near future. Okay. With that said, I would think about what's the research priority as we talked about it. So it's kind of the very important 6 point. I will start with the patient community. Now, as we learn a lot from the patients and what their needs, and I like pretty much the idea of the qualitative research that's just discussed before this talk and the focus group. And I think we can learn so much from these kind of open-ended questions and discussion with patient. And also we learn from the patient what their most important needs, and that will direct us on the next few years on how we can make a goal and make a target for us as a group for researcher and the patients, and partnership with the pharma, and looking for funding also. So as we conclude, I think in my mind, when I look at this, I think it's this kind of a shared vision between the researchers, between the patient, and we have all this ultimate goal to try the best as we can to move the research forward, to learn about the conditions in terms of the basic, and also to look for valuable resources, valuable model, and look for undetermined or undefined biomarkers that can help us in the future to develop a transformative therapy for this condition. With that said, I would like to have a huge thankful to the whole group. Without the funding from the Global Dare, ULF, the GFPD, RhizoKids, I wouldn't pursue this skills and learn so much about this condition. So I really grateful for that, and I'm grateful for the community to learn from, all the groups, the huge group that the Kennedy Krieger, my mentors, my supervisors, and the whole community. Thank you so much, and I think we can be happy to open this for discussion.